Great. Let's get going again. I'm very flattered that you're all still here after listening to my voice for 2 hours. So we're going to talk about the genome, and I'm going to continue this on Friday morning with a slightly different part of it. So what I'm going to talk about, these are what I'm going to cover in this series of 2 lectures. So hopefully in the past 2 hours we've kind of given you the background of DNA sequence variation, how we look at it, what it means, the ways that you can get a single gene disorder like sickle cell anemia or thalassemia, all the different things that can go wrong in the DNA. We're going to talk more globally about the whole genome now and how that evolves and varies and what that means, and in particular on Friday, how we're using this to attack much more complicated diseases. Pretty much everything I've been talking about so far applies to single gene diseases, what we often call Mendelian disorders, where it's a single gene that has something wrong with it. And, you know, that's sickle cell anemia and thalassemia and hemophilia and BRCA1. There are lots of, there are probably about, now I think the catalog's up to about 3,000 or 4,000 diseases we know of due to a single gene disorder. But there are lots of other diseases that are very common that are much more complicated. It's not one single gene. Diabetes, heart disease, most of what one sees in practice, in a general medical practice. And that's going to be the subject of Friday. But I want to talk about today is a little bit more, we've talked about some of this. I'm going to re-emphasize and go into some more detail about what actually makes up our genome and how it's put together. We're going to talk about recombination, which is a very difficult. We're going to talk about in this part what I think are the 2 most conceptually difficult things that we're going to cover, maybe in the whole this whole course, but certainly in my lectures. But I think they're important concepts to have a general feel for. What recombination is, what linkage is, what linkage disequilibrium is. And hopefully this makes, well, I'm guessing that this makes absolutely no sense to you at all now, sounds like complete gibberish, and I'm hoping at the end of this next hour, 45 minutes, this will sound at least vaguely familiar. Okay, so we already talked about all this. DNA sequence variation, there's a lot of it. 0.1% between any 2 humans, 1 to 2% if we compare ourselves to chimps. You all are familiar with polymorphisms. You understand this distinction. This is really, really important. Any change in the ancestral DNA sequence is a mutation. The millions and millions of variations we all have all arose as a mutation at some point. Everything different between us and chimps is a mutation, either in us or the chimp. Anytime sequence is different, it arose from a mutation. Mutation is not synonymous with bad, causes disease. Without mutation, we would all still be bacteria or something in the primordial soup. So mutation is not bad, though it's commonly used that way. This is a very common misuse to say, when we do this BRCA sequence in our patient, is this a mutation or is this a polymorphism? The idea being polymorphism benign, mutation is bad. That's wrong. Polymorphism just simply means it's common. And mutation, everything's a mutation. What you really want to know is this a disease-causing variant or a non-disease-causing variant? Is it common, a polymorphism, or not? Okay. And then we talked about how to re-emphasize, to beat this point a bit more, a normal change, if it's very common, is a polymorphism. If it's rare, it's a private polymorphism or a rare variant. If it causes disease, it's a disease-causing variant or disease-causing mutation. And if it's common, it's still disease-causing, but now it could be a polymorphism as well. Okay. So this is a little bit of history, but I think it's informative. And what I'm going to talk about now is linkage. And this is less important than it used to be, but it's an important concept, I think, for you to understand. And I spend much less time talking about this than I used to because it's becoming more historic. But I think you really should understand the concept. This is basically the idea that different parts, if you look in our genome at genes or parts of the chromosome that are near each other, they tend to get inherited together. And so we can use inheritance of some marker in the DNA as to keep track of some other gene that's causing a trait. And this is actually the first example of this from 1968. And this was a heteromorphism on chromosome 1. So this is, I was telling you, used to do cytogenetics. We still do it somewhat. It's becoming used less often. Take a cell, spread open the chromosomes during metaphase, stain them, and you can look at them like this. The largest chromosome is chromosome 1. The next largest is chromosome 2. And chromosome 22 is the smallest. And then you have the X and the Y. We just numbered them based on size. And this is what they look like. Well, Donahue observed that if you looked at people, a lot of normal people, you found some people had this funny thing on chromosome 1. It's part of the DNA is just sort of, it's heteromorphic. It's sort of, it's spread out a little bit. What's that about? And he observed in this family that if you had this heteromorphism, that's the people colored in yellow, this got inherited along with the Duffy blood group, which is a blood group, that a subtle difference, common polymorphism on the surface of red blood cells. That's a place, by the way, where polymorphisms are really important because ABO, for example, is a common polymorphism on your red blood cell. If you have type O, you're type A, you're perfectly fine. But if you're type O, you will have antibodies against A or B. And if we give you a transfusion from somebody who's type A, you will have a transfusion reaction, which could be fatal. So when we give a transfusion, we just don't give a transfusion to anybody. We have to match it for a whole bunch of polymorphisms. That's a place where being polymorphically different is, even though they're not disease-causing, it's really important in the way we do medicine. So this is one of those blood groups. And look at this. If you look at, this guy has Duffy A and Duffy B, and both of his, and mom is BB, both kids must have gotten B from mom, so they had to get A from dad. And they both got the heteromorphism. And this child got A from dad, because he's AA, and also got the heteromorphism. Basically, everyone in this family who got the Duffy A allele from a parent who had the heteromorphism got the heteromorphism along with it. They're being inherited together. That's linkage. This heteromorphism is linked to the Duffy blood group. Does this heteromorphism cause the Duffy blood group? What do you think? Nope. We can prove it because we can find a lot of people who have Duffy A who don't have the heteromorphism. So you don't have to have the heteromorphism to get the Duffy A blood group. But within this family, they're traveling together. That's what linkage is. Okay. And why is it? Why does it occur? It occurs because of this phenomenon of recombination. During meiosis, when we make an egg or a sperm, we line up together the maternal copy of each chromosome and the paternal copy of each chromosome during meiosis. And they pair up very closely. They form something called the synaptonemal complex, where the two strands are right next to each other. And exchange occurs where you shuffle a little bit. A bit of dad's chromosome 1 gets swapped for mom's chromosome 1 by recombination, just like what we showed in the aphaglobin example where you got a deletion. That was a wrong recombination. But normally the sequences line up perfectly, and you just exchange from one to the other. This happens all the time. It happens on average in one meiosis about, in one set of chromosomes that you get from your mom or dad. On average, it's at least once per chromosome. Typically, this is happening. So if we look at you and we look at your mom or your dad, the chromosome 1 you got from dad, you'll have one chromosome 1 that came from dad, one that came from mom. But the one that came from dad will have part of one of his mother's chromosome and part of his father's chromosome, and the same for the one you got from mom. We're always a little bit of a shuffling. This is another way we generate diversity among us during evolution. So this is going to occur at least once, sometimes a couple of times along the chromosome. Now, imagine that you have two different genes that are fairly near each other on the chromosome. Okay? So depending on where that recombination occurs, so let's take this example where the gene is sitting, they're fairly far apart. This chromosome has big A and big B. This chromosome has little a and little b. They're fairly far apart. A recombination has occurred in the middle. Okay? So we end up with these chromosomes. You either got the part that didn't recombine, big A with big B, or you got a recombination, you got a chromosome that's got big A and little b, or you got the other one that's got little a and big B, or you got the little a, little b one that didn't recombine. Does everybody follow this? Well, What if A and B are really close to each other and the recombination occurred above it so that they got swapped together? Chromosomes look exactly the same. You still have these mixed chromosomes, but if we look at the genes, A and B, big A and big B, and little a and little b always stay together. Okay? That's linkage. And generally what happens is the farther apart the genes are, the more likely they are to recombine during meiosis. And the closer together they are, the less likely they are to recombine during meiosis. Does that make sense? This is a very tough concept, but it's a really important one. It turns out, we know, that the frequency with which this happens from years of genetic studies is roughly the chance of two genes recombining with each other. The chance of the recombination recurring between these two genes is roughly 1% for every million base pairs. So if two genes are 1,000,000 base pairs apart, 1% of the time they're going to recombine. They're 2 centimorgans apart, 2% of the time they're going to recombine. If they're 50 centimorgans apart, 50% of the time they're going to recombine. What's the most they could ever recombine? 50%. Yeah. Because if you know, it was more than that, you end up coming back to the other thing. 50% is the maximum you approach. And that's the furthest. If you do two opposite ends of the chromosome, the most you can do is recombine so they get totally randomly scrambled, and it's 50%. Then they get the two ends of the chromosome are far enough apart, they get inherited independently, just like two different chromosomes get inherited. So it's not an absolute thing, but across the genome, there's some places that recombine more than others, but statistically, on average, it's 1% per million base pairs. Okay, so let's take a look here. Here we have disease D. Could be any disease. And we have a marker, a gene nearby, a SNP, a polymorphism, the common one nearby, that you could have either big A or little a. And in this particular case, the disease mutation in this father with the disease, an autosomal dominant disease, occurred on a chromosome that happens to have the big A. Now, every child is going to get a big A and a normal copy of the gene from mom, and they're either going to get a big A or a little a from dad. And you'll notice that these three who got the disease all got the disease gene from dad, and they got the big A. And these three individuals got little a and the normal copy from dad and didn't get the disease, right? It's linkage between this gene and the disease. Well, here's the exception. What's going on here? This person got a little a but got the disease because there was a recombination between the two. Okay? The closer together, the less often this recombinations occur. The further apart, more often. So we can use this kind of polymorphic variation to track the inheritance of the disease gene in a family, even if we have no idea what the disease mutation is, even if we have no idea what the disease gene is. As long as we know it's a gene, and we know this marker is near it, we can use it to trace the disease by linkage. Okay. And in fact, this is how we used to find disease genes when we had no idea what the disease was, what the gene was. This is neurofibromatosis and a DNA polymorphism. This is one of those old RFLPs by Southern blotting. Doesn't really matter how you do it. The point is, it's a common polymorphism. You have either this big band or you have the little band. In all normal people, you could look at anybody and that's what you would see. In this particular family, dad, who has the disease, has one of each. Mom has two small bands. We look at this kid, he had to get a small from mom, in which case he got the big from dad. This kid got the big one from dad, this kid got the little from dad. And if you look, you'll see that, with one exception, I think, everybody here who got the dad's big copy got neurofibromatosis, and they got the little copy, they didn't get neurofibromatosis. Out here, there's one exception. So this guy got neurofibromatosis from dad, has the big copy and the little copy, right? And here, the mom, who's normal, has two big copies. Now, important thing to remember, whether it's big or little doesn't mean is not the disease gene. Here's somebody who has two bigs and is normal. Here's somebody who has two littles and is normal. That is not the disease gene. It's just what's nearby. So in this guy, again, it's the big copy that has the gene, and everybody in this side of the family is going to get a big from mom, and it's whether they get the big or the little from dad is going to determine whether they get neurofibromatosis. Here's the one exception, this individual who got the small copy, what should have been the normal copy from Dad, and also got neurofibromatosis. How did that happen? Recombination between the marker and the disease. And out of all these individuals, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Out of these 17 individuals, this happened once, a little over 5% of the time. So this turns out this is about 20 million base pairs away, and we expect it to happen. It's about 5 million base pairs. We expect it to happen about 5% of the time. Yeah?
So if they were closer, there'd be more recombination events?
No, closer, there'd be less recombination events. If they're further apart, right? Make sense? Okay. So this was the way positional cloning became possible. You could now take a disease where you knew absolutely nothing at all about what caused the disease, and simply, if you could have a set of these markers that covered all the chromosomes, just look at a family like this and just see, of all the markers we're looking at, which one tracks best with the disease in the family. The gene, whatever it is, must be near there. And that's what positional cloning is all about. Here is the first successful mapping of a human disease gene this way. And this is a disease called Huntington's disease, autosomal dominant. If you get this gene, you get the disease. We had no idea at all what the gene is. You've heard about this, I think, or you will. This is a terrible neurodegenerative disease. You're perfectly fine and healthy until you're in your 30s or 40s. Then you get this rapidly progressive movement disorder, dementia, and you die. Not so great. When you're 20-something, it doesn't seem so bad. When you're my age, it seems really awful. But it's bad. And living your whole life, not to make light of it, living your whole life knowing you've got this hanging over you is obviously pretty terrible. It's a very significant disease, and we can diagnose it perfectly now. The gene wasn't known at the time. And it's a very common in a particular village in Venezuela. And this is a family from that village. And this is a polymorphic marker that happened to be on chromosome 4. This was from, you remember I showed you that slide of the markers we knew of on chromosome 4. They only had 3 markers on chromosome 4. They were incredibly lucky that one of them happened to lie right near that gene. Amazing luck. That's where their luck stopped. They weren't able to find the gene for another 10 years. But they were able to figure out where it was. Because if you look at this family, and I encourage you to go through this on your own with the slides, you can trace the inheritance of where this is being inherited, which allele this is being inherited with. And this allowed us to know that the Huntington's disease gene, it was segregating almost perfectly with this marker, which was G8, the 8th marker that was identified. The chances working were, I remember when I heard about them trying this, this couldn't possibly work. You need hundreds of markers to do that. And at the time, to have 300 or 400 markers in the human genome seemed impossible. Now we have 25 million. At the time, it seemed, no, we'll never have that many. And they only had 8, and they tried anyway, and they were so lucky that this one happened to fall right next to the gene. So we knew the gene lay on this part of chromosome 4. It meant you could do this typing for this polymorphic marker that's got nothing to do with anything, except it happens to be near the Huntington's disease gene, and you could predict accurately who was going to get Huntington's and who wasn't. So the first thing you would do from this approach, by checking markers throughout the genome and a family, just seeing what got inherited along with the disease gene, you could figure out where the gene was. And then you had to do all kinds of really complicated stuff that you don't want to know about because it's ancient history. How many of you can use a slide rule? I didn't think so. This is like a slide rule. How many of you have heard of a slide rule? Okay, that's makes it feel a little better. But, who would ever use that kind of thing now? There was a time when this seemed miraculous in its ancient history. Now we do it all in silico. Once we know where the gene is, we can sit down at the computer and look at all the genes that are in that area and figure out which ones it might be and start checking them until we find the actual mutation, and bingo. Everybody who's got this disease has a mutation in this gene, and we know we're there. Okay. Cystic fibrosis was the first gene that was found purely by this kind of approach using this kind of genetics. It was a huge splash. One of the major leading groups was headed here at the University of Michigan by Francis Collins, who's now the head of the NIH. He used to give this lecture, by the way. I had to take it over from him when he left. I would have, as much as I love giving this lecture, I wish he'd stayed and give them. He used to come back for a while and give them, but anyway, the cystic fibrosis is now routinely screened for. I was mentioning this to somebody earlier. Women who go to the obstetrician, this is now offered as standard of care if you'd like it. 1 in 25 people of Europe. It's found in all human populations. It's particularly high in Europe, where it's 1 of 25 people are carriers. Again, probably 8 or so people in this audience are carriers of CF. It's an autosomal recessive disease. Being a carrier, like sickle cell, is fine. It's only when you have 2 copies that you have the disease. But we would have had no idea what that, we had no clue as to what might be causing that until this methodology made it possible to isolate the gene. And this was an enormous effort. I actually pointed out, I think on the Huntington's one, both of these were efforts of huge consortiums of thousands of people and tons of labs around the world working on these things to do it. And it's now become almost routine, as I'll show you in a minute, easier than routine. In fact, we don't even bother with most of this anymore. We talked about the different types of variants you can use. I talked about these microsatellites or simple tandem repeats. These were very useful for linkage. And maybe you can see why now, because you have so many different alleles. Everybody, most people are different if you look at this thing. There's so many different bands you can get. If you could find 300 or 400 of these spaced evenly across the genome, like this, you were ready to do this. All you needed was a family. And if you had a family that had the disease, you do this and you could find where the gene was. And that's what we did. And that was what genetics was all about for the past maybe 10 years. When we found the gene, then you had to look for mutations. And there are, we talked about the different types of mutations. Most are silent, et cetera, all these different variants. We talked about the fact that we now have many millions of these variants. This was hard to do in the days of Huntington's disease gene. Nowadays, you can take a little SNP chip for a few $100s. You can check 1 million SNPs throughout the genome and everybody. And we don't even bother with STRs. This is how everything's done now with these common single nucleotide variants. The next huge quantum leap forward for us was the sequence of the human genome. And this used to be big news. This is now 11 years old. Not such big news anymore. This became possible because of sequencing technology. I told you about how when I started out, I spent 1.5 years sequencing 4 nucleotides. Well, these automated machines like this is one of the early genome centers, could sequence millions of base pairs in a day. This was thousands of people working hard. You could do millions. It still took a long time to do the whole genome. Nowadays, as I told you, we could do billions in my lab, one person. It's not so hard. And at a fraction, teeny fraction of the cost, and I'll show you that in a minute. Anyway, this made it possible to sequence large amounts of DNA and to sequence the entire human genome, something that when I was a medical student, we knew that DNA was a genetic material. We knew there were ACs, Gs, and Ts, but we actually weren't exactly sure how big the genome was. We were totally mixed up about that. The idea, if you would have told a group of scientists then that someday you'd have the entire human genome to look at, they would have laughed you out of the room. It's impossible. But it's been done, and it's totally astounding. This was the article of Nature in 2001 when we announced the genome was done. It wasn't really done, but it was almost done. And this has totally exploded the way we find disease genes, completely revolutionized it. That work, the cystic fibrosis gene, thousands of people in many labs, hundreds of labs around the world, working night and day for multiple years to find the gene. Now, it's an afternoon in a good lab if you have the right DNA samples. And you can see what happened when I was in medical school. The number of diseases at which we knew the cause at the molecular level, the responsible gene, was 1. Sickle cell anemia and thalassemia. Look what's happened. And this is where the human genome came off, and things just really took off. And this stops at 2005 because it's exploded since then. I'll show you on a later slide. We're now up to about 5,000. So basically, the bottom line is now, if we have a human genetic disease, a single gene disorder, and we have a large enough family, and even that isn't required anymore, where we can see how it's being inherited, and we have DNAs on those individuals, we can find the gene and identify what the gene, in a research lab, identify the responsible gene and figure out, and then precisely diagnose the disease in individuals in that family. Yeah, and other families. Yeah. So the question was about multifactorial diseases like type 2 diabetes. It's impacted it in a huge way, though it's different and it's been much harder than we thought, and our progress there is much, much, much less. its impact in the clinic right now, I would say, is somewhere between 0 and none. But that is the entire subject of my next lecture. So we'll talk about that. Okay. So we go from having the genome to having knowing where the gene is, to finding the single gene that finds it, that causes the disease. We have this range of genes from one of the littlest, the globin gene, that's about 1,500 base pairs, to one of the biggest, dystrophin, which causes muscular dystrophy, 2 million base pairs. And we can figure out how many genes we have. And this was a cause of great consternation to many people when we actually looked at the genome, because we had all been taught for years and years and years, based on old, very definitive experiments, that there were 100,000 genes in the human genome. Well, it turns out we only have 20,000. And many people were profoundly upset by this, because earthworms also have almost the same number of genes, and so does the fly. And most people found this really insulting to our species. But we've learned to live with it. The fact is, we're really, in terms of the number of genes, we're no more complex than a fly or a worm. Now, we seem to have more complex splicing, and many people take comfort in that. They think, well, okay, we don't have more genes in a worm and a fly, but ours are more complicated, and they do more things. But I'm not really sure that's entirely true either. We don't even have all that much more than a yeast or a bacteria, fair bit more than a bacteria, which is really kind of astounding, because it certainly seems like we're more complicated. Even having the number, we have a fairly big genome. It's bigger than some of those species, but plants have bigger genomes than we do. And that's really distressing to some people. But we still like to think we're better than all those other species. We now have, this is out of date. It's recently exploded out of date. We now have complete human genome sequences probably on about 10,000 people. So I was just off by a couple of logs here, because this is changing so quickly. It'll be millions soon. We have, this is also way up, but we have now the complete sequence. Almost any species anybody's interested in, you just do the whole genome now. We do some experiments in my lab where we look at bacteria and mutants of the bacteria. And if we want to find the mutation in the bacteria, the easiest way now is just sequence the whole bacterial genome. It's why bother looking for the single change? Just sequence the whole thing and see what's different. The problem is, bacterial genome, which is only about 5 million base pairs, that's so tiny that it's a waste to put that on the machine by itself. So we'll usually pull together 10 or 20 different ones so we can do them all at once. So it's really amazing how this has exploded. So what do we know about the human genome? What have we learned? Human genome, as you know, is 23 pairs of chromosomes, a total of 46 chromosomes, which the genes are spread across, a total of 3 billion base pairs. That's the haploid genome. So the DNA you get from mom, you get 3 billion base pairs from mom, 3 billion from dad. A total of 6 billion, but that's duplicated. So it's really 3 billion unique in the haploid genome. A lot of it, most of it, is repetitive sequences, control regions, spaces between genes. We have no idea what most of this does. Really no idea. Some people claim they have an idea. We don't really know what's going on. About 30% is where the genes are. But even the genes, most of it is space. It's the introns. The introns are where the dystrophin gene is 2 million base pairs. Only 20,000 of that codes for protein. The other 980,000 is introns. So most of this is intron, space in the gene, most of which we don't know what that does either. The total, so of this, again, only 1.5% is actually the coding sequence. Now, if we look across all the species, if we take our sequence and line it up with chimps and dogs and cats and mice and worms and flies, we find a very highly conserved region pieces, including all the genes. The exons are pretty highly conserved. They really stick out. They're much more the same compared to other. There's a protein we work on in our lab, and we were just astounded the other day, and we looked. I mean, we should have known this, but it's a part of the ER, and this protein in humans is 50% identical to the protein in yeast. That's a long evolutionary distance. So we can't. If you look at an intron, you won't find any similarity at all to the yeast. There's another 1 or 1.5% of the genome that's just as highly conserved as the exons. We have no idea what it does. It must be doing something important if it's that highly conserved, but it doesn't code for protein that we can tell, and we're just starting to figure out what all that does. So there's still lots to do. So if any of you are actually interested in working on this stuff in the lab, there's still plenty of questions to answer. The sequence now, it covers almost all. There are a few parts of genome that have been very hard to sequence and hard, so it's not really totally complete. And there still are some errors, which people kind of assume if it's in the genome, it must be right. It turns out that its accuracy is 1 in 100,000. There's less than 1 in 100,000 errors, which is astounding. In the old days, we sequenced. If we got 99% accurate, we were thrilled. And this is the whole genome's been done at this level. But that means that there are still some mistakes in there. In 3 billion base pairs, that's still 30,000 mistakes. And often, it was a big issue. Whose genome do you sequence? It was actually a pool of genomes, so you couldn't identify the person. But what's normal? So factor V Leiden, I told you about this variant. We study it in my lab. 5% of Europeans have it. Causes an increased risk of thrombosis, but you still do pretty well. 5% of people have it. Turns out the normal sequence, we just noticed this the other day, the normal sequence in the genome has factor V Leiden. Even though it's only 2.5% allele, it's a polymorphism, but it's the rare allele. And in many cases, it's the rarer allele that's in the index genome. Important thing to keep in mind. There are still a few holes. This is less than when I made the slide. Wonderful thing, this is all freely available to anybody. If you'd like to read the entire human genome, you can just go to NHL Beyond. You can sit there. It'll take a while, but you can read through the entire thing. And there are browsers and all that make it actually mean something. I told you already, because I'm still wrestling with this, that we have fewer genes than we thought. And we like to think that we make more complicated proteins. And I already talked about this. So these are the base pairs in GeneBank. This is the repository at the NIH, part of the National Library of Medicine, where all the sequences you want to look at, you can just freely get to with a web browser anytime you'd like. And you can see it's grown exponentially. And I forget where it is now. It's actually, this is a terabyte, 1,000 gigabytes, and we're actually way over that now. I think it might even be getting close to petabytes. That's the next thing after terabytes. That's 1,000 terabytes. It's unbelievable how much sequence there is. This is a wonderful place to go. If anybody's interested or excited about this, I would suggest you sit down and you go to the NCBI website. This is the part of the National Library of Medicine. This is where GeneBank is. You'll know these guys also because they also run PubMed. For most of us, is the most important part of the NIH. Because PubMed is extremely useful, and this is for DNA, obviously what PubMed is for everything else, for the literature. And there's lots of things you can read about here, and you can look at, and you can look up any gene you want, and it'll tell you all sorts of things about it. There are other web browsers. This is the one at UC Santa Cruz that many of us use, that researchers kind of like. You can go in there, look at your favorite gene, and this is one of mine. As a gene we work on in our lab that causes a disease called TTP, which we found by positional cloning ancient times, 10 years ago. Things had already improved by then. I told you how cystic fibrosis with thousands of people working on it night and day for years and years. Adam TS13 was a Michigan MD PhD student working in my lab for 9 months. Now, she could probably do it in about 2 weeks because of technology. So it's gotten incredibly fast to do this. And this is the Adam TS13 gene. And you can see all the other genes around it and what parts are conserved compared. You can look at all sorts of things. This is a genome browser. Extremely useful, mainly for research purposes at this point. Okay. So we already talked about this linkage concept, and I'm just emphasizing again here because this is so fundamentally important. Linkage in this case was because this A and B gene tended to be inherited. Whatever genotype you had at A and at B tended to be inherited together. That's what that's linkage. I'm going to talk about and this is now the most difficult concept that I'm going to tell you about. This is linkage disequilibrium. Related to linkage, but something a little different. And I'm going to spend some time talking about it. If you don't fully get this, don't feel too badly about it. But it's a difficult concept, but I think it's important. Now, what I'm showing you here is the HLA locus. This is one of the most polymorphic parts of our genome. This is the histocompatibility locus, the major histocompatibility complex. It's highly variable. So unless you're related, unless you look at your siblings, most of us have different HLA types than other people. And this is part of how our body's immune system tells self from non-self. And that's why if we put a skin graft on you from somebody else, you'll reject it. Or if we give you a bone marrow from somebody else, you'll reject it. Because your body says, oh, wait a second. That's not my HLA type. That's foreign, and it gets rid of it. And so if we do a transplant, we type. We try to get a match for HLA. So it's a very important medical thing, but it's incredibly polymorphic, and there are a whole bunch of genes in here. One of them I'm going to say a little more about turns out to be a gene called HFE. It's a gene that causes hemochromatosis, which is a very common human genetic disease. So here's HLA typing, and this is the old way that we used to do it, but I think it's instructive. So there's a bunch of HLA genes in here, A, B, and C, and the D types. And you can type for all of them. And there are many different types. You could be A1, 2, 3, 4, 5, 40 or 50 or so different alleles at each one of these. So here's a family. And we've typed dad and the kids. Okay? So dad has, at A, he has two alleles, A1 and A29. At B, he has B7 and B8. At D, he has DR3 and DR4. Okay? And here's the types in all the kids. Now, he's got two chromosomes. And we don't know whether A1 goes with B7 or B8 or DR3 or DR4. We don't know how they're lined up. That's called phase. This is his genotype. Knowing which ones are on which chromosomes is phase. You can figure that out. So here's how we can deduce that. So if we look at daughter number 1 here, okay, she's A2, A29. Well, which one did she get from dad? A29, so that's here. She's got B7 and B35. Which did she get from dad? B7, so etc. So we can tell that this chromosome came from dad. So that's told us one of his chromosomes, okay? And by deduction, what's left over is the other chromosome of dad's. And by deduction, her other chromosome must be one of mom's chromosomes. So by that kind of logic, and again, I would encourage you to go through this on your own with the figure in the handout and figure out and make sure you can derive this from this. These are genotypes. This is their phage. What's actually on each chromosome? And this is extremely important when we're typing patients to figure out who do we want to use for bone marrow transplant. So let's say this kid has leukemia, and we want to use one of her siblings as a transplant donor. Who would you pick? 4 or 5, you might say, right? They got the same chromosomes as she did from dad, except look at this one. A2, B35 is supposed to be DR13. It's DR1. How'd that happen? What? Recombination, crossing over. Very good. Exactly. In mom, it just so happened there was a recombination right here, and DR1 ended up here. So this kid is fairly close, but one part of the HLA locus is different. So this is a better match. So that's the person we'd use for the transplant. This is a perfect HLA match. This person is haploidentical, has one chromosome the same. This person is completely non-identical. By chance, each of your siblings has a 1 in 4 chance of being HLA identical, a 1 in 4 chance of being completely different, and a 50% chance of being half identical or haploidentical. Okay, well, here's a curious fact. Hemochromatosis. Fairly common genetic disease, 1 in about 800 or something. Roughly 1 in 8 or 9% of people are carriers. A fairly common disease. Curious thing, if you look at people with hemochromatosis, 70% of them are HLA-A3. 30% of them have other HLA types. How could that be? In the general population, only 10% of people are A3. So does A3 cause hemochromatosis? No, because you could have 2 A3s and not have it. Do you have to have A3 to have hemochromatosis? No, there are plenty of hemochromatosis patients who don't. So what's going on here? This is linkage disequilibrium. What has happened here is there's a founder effect here, like the sickle thing where there were 4 different mutations in history that caused sickle cell anemia. In the case of hemochromatosis, there was one founder. That mutation, when it happened, by chance, the new mutation in that individual, just happened to happen on a chromosome that had A3. The HFA gene turns out to be very close to HLA-A. So they tend to be inherited together. Recombination between them is very rare, extremely rare. So what happens is, even though this happened 20,000 years ago, over all that time, they're still being inherited together. Not always, there have been some recombinations that separated them. They're still being inherited together. Now, if A3 was located a long ways away, we would have expected everything to have been reshuffled by now. You would expect the HLA types in hemochromatosis patients to be identical to the ones in normal chromosomes. That would be called linkage equilibrium. What's happened is there hasn't been enough time yet for this to get all shuffled, and there's still a big excess of A3 in HF with hemochromatosis. So this is called linkage disequilibrium. Now, it seems like an obscure fact. It is somewhat obscure. It's going to become important on Friday when we talk about complex diseases. And I'll re-emphasize at the end. If it's still, I'm sure this is very confusing. I've tried for years. I've still never figured out a good way to make this really straightforward, and would love suggestions. If you're still a bit puzzled, don't worry about it. We'll go over it again on Friday, because it will help us understand the idea of, and I hope it'll get more clear then, of how we look at complex diseases. The extension of this is laying the groundwork for Friday. Let's take a look at some SNPs. Here's 3 SNPs, common SNPs, variants. You can have either a C or a T here. You can have either an A or a C here. You can have either an A or a G here. Okay? How many possible combinations and permutations are there? Well, 2 times 2 times 2. You think 8 ways you could put all these together, and these are all 8 possibilities. Well, guess what? We go look at everybody in the room here. What do we find? Only these. This is another example of linkage disequilibrium. And the reason, again, is probably when this variant occurred, this isn't a good example because these are the same. The various changes that occurred, this T to C occurred on an allele that had this. There obviously have to be. You can't observe only these 2. There are others. But these are the most common. It has to do with the history of how the mutations occurred and where they occurred and what chromosome they occurred on. And they're still in linkage disequilibrium. This turns out to be a phenomena throughout our genome. Part of what's happened is this phenomena here. There were many humans, well, not so many as we have now, but in Africa, when we left Africa, those of us who left Africa, a limited number left. And this is true anytime a human population moves. If there's a limited number of founders, you could have an allele that's very rare here. If it happens to be present in one of these individuals, as that population expands, it'll be much more common. So human populations around the world have somewhat different frequencies of these various polymorphisms. Most all of them arose back here. You'll have different frequencies, but they're looking still at the same ancestral chromosomes. And since this is all fairly recent in evolutionary terms, there hasn't been enough time for everything to get shuffled. If 2 SNPs are sitting a few thousand base pairs away from each other, there hasn't been enough evolutionary time for them to reach linkage equilibrium. So they're tending to be inherited in blocks. And what we find when we sequence lots and lots of genomes is there are blocks. The average in size is about 20,000 base pairs. Large blocks that tend to be inherited together. Just like the hemoglobin, the HLA-A3 in hemochromatosis example, it's not perfect. These blocks do get broken up, but they tend, if you have an A here, you tend to have a G here, if they're really close. And the genome gets inherited in blocks like this. And this is laying the groundwork for what we're going to talk about on Friday, when we're looking for common variations causing common diseases like heart disease and diabetes. We're going to look with these blocks. We're going to say, are there any particular blocks that get in here? We don't know what the change is in that 20,000 base pairs, but if this block is more common in people with diabetes, there's probably something somewhere in there. And this is all based on linkage disequilibrium, that these blocks have not shuffled enough to all be random, that parts of the genome are still tending to be inherited together. So what we're going to do is look at a bunch of people with a disease and look at a bunch of unaffected people, and if they're somewhere in here on this chromosome, a mutation occurred that causes, let's say this is the region around the A3 gene of HLA, and a mutation occurs right here in the HFE gene. When we look many years later at people with hemochromatosis, we're going to find much more of the red. Then it'll still be in the unaffected, but at a lower ratio. And this is, again, because of linkage disequilibrium. Now, all we know is that the responsible gene and mutation is somewhere in this big block, this haplotype block, this block of linkage disequilibrium. But we still don't know what it is. This is how we made this deduction that I told you earlier about sickle cell anemia. It turns out, if you look around the hemoglobin gene, and all people with sickle cell anemia, there are basically only 4 different colored blocks around the hemoglobin gene. And everybody has one of those 4 colored blocks, one of those linkage disequilibrium blocks, one of those haplotypes. And they're associated with these 4 different occurrences of the same sickle cell mutation. And we can now trace it over evolutionary time. Again, we just take everybody with hemoglobin S, with sickle cell anemia, and we're going to find a different distribution of these haplotype blocks than we find in the rest of the population. And that's this evolutionary history. And then using this in a more general way is, again, what we're going to talk about in the next lecture. I've talked about how the technology has changed, astoundingly. We can now sequence many millions of base pairs in a day. And that's what this next-gen sequencing has totally revolutionized what we're doing. Not only made sequencing, the human genome was done without this next-generation massively parallel sequencing that we're now doing. It was done with these old-fashioned machines. These newer machines are dramatically, orders of magnitude faster and cheaper. This is what's making it possible for us to sequence thousands, tens of thousands of genes, and eventually all of our genomes. And we're going to talk about the implications of that for medicine, genome-wide association studies for human disease. That'll all be the focus of my last lecture to you on Friday. So stop there. Thanks for toughing it out for these 3 hours. I'll hang around if people have any questions, and I'll see you Friday.